Distinguished Alexander, distinguished colleagues, I would like to welcome all of you at the second day of the symposium dealing with orphan genetic diseases, the current status on family hypercholesterolemia. Allow me to give the floor to Professor Katapana, who has connected us online for joint convening of the session. Professor Katapana.
Hello, everyone. And welcome to this session, which will be on the modern diagnostic methodologies and gene therapy for related lipid metabolism disorders and viewpoints of pharmacologists and clinicians. It's a pleasure to chair this session together with Dr. Alieva. And without any further ado, let's start with the first presentation. It will be by Professor Kostareva from St. Petersburg. She will be talking about gene therapy, new opportunities in cardiology. Please.
I will start with a basic presentation section that will consider the possibility of gene therapy in cardiology. First of all, I would like to highlight on the aspects that concern the possibilities of current gene therapy in clinical medicine in real clinical practice. Some time ago, it seemed to us that gene therapy is absolute theory that would be impossible, and it was not clear from the viewpoint of safety and ethics. Now these drugs are used in clinical practice, not only cardiology, and of course, this is primarily oncology, neuromuscular diseases, and then other areas of clinical medicine, including cardiology. So today, I would like to cover the aspects that determine the possibilities of using gene therapy in different fields of medicine. These possibilities are associated with the gene size. The larger the gene, the more difficult it is to work with it, to deliver it to the tissues and cells and the diseases. When we have to do with very large genes, and with a large volume of genetic material are not suitable for gene therapy, then the target organ or tissue, as a rule, in most cases, that is a system injection, and we have to ensure tropism, arrival of therapeutic construction to the area which we need, or to work with those nosologies where there is a lesion of most of the organs and tissues and where tropism is not as important. So one of the areas and trends were systemic diseases, metabolic diseases, disorders of metabolism, where the lysosomal and mitochondrial enzymes are working concerning all tissues and organs. It is necessary to overcome the barriers, for instance, the natural barriers and blood-brain barrier, because it is not overcome. Work with tissues of the nervous system, sense of the nervous system is not possible. For a long time, a very complicated issue was that of safety. Most often, gene therapeutic drugs were developed on the basis of viruses. Viruses are not always safe, well integrated, and until significant data are accumulated about the safety of viruses, that question was open. And the last area that is probably not so fundamental, but we have to take into account, gene therapy and the development of these drugs is very expensive, and it has inherent risks. So it is justified in case where a lot of therapeutic approaches, pharmacological, surgical approaches are exhausted, and there is no other possibility to affect on the target gene. Gene therapy can be used in monogene pathology. That's how it started and initiated. In polygene pathology, for instance, ischemic conditions of lower extremities, diabetes mellitus of the second type. Most often it is used in orphan genetically caused diseases. There are several approaches. First approach is mostly replacement of the defective gene. Due to mutation, the gene doesn't operate and the protein is not synthesized, and there is a lack of function of the protein. These are the so-called diseases of autosomal recessive character, and adding the gene will supplement the deficit and cope with the adverse clinical situation. These are most of approaches in case of orphan genetic pathology, and in certain cases inherited hypercholesterolemic. In polygene pathology, we sometimes add an excessive copy of the normally functioning gene. For instance, growth factor of vessels that increases vessel formation. It might be added in acute ischemic conditions in case of infarction. These approaches were developed. They are not actively used in clinical practice, but they are still used. There is an approach with gene suppression when we have a mutant gene and there is a synthesis of protein that has toxic properties in respect of cell, that is an autosome dominant character of inheritance. If we want to suppress the protein that adds additional functions, we use the suppression of the gene. And the last option, editing of the genome, when inside in the organism or in human cells, taken outside but then returned to the human body, there is a change in genetic material of certain genes. That is a risky approach that is very complicated in terms of technology, but using CRISPR-Cas technology, it exists in practice. A very difficult use of gene therapy that restricted it was the question of delivering, how to deliver the genetic material that we want to deliver to a certain place. Most of the cells of our organism are thermally differentiated. The more differentiated the cell is, the more difficult it is to approach it. Muscular cell, myocardium cell, and vascular cells are terminally determined. It is difficult to introduce genetic material, unlike the proliferating epithelial cells and skin surface cells. We need very aggressive carriers, for instance, viruses. The second option of delivery, non-virus mediated delivery, we have to use certain carriers that will easily get into the cell, pass through its membrane. This will not be a receptor-mediated admission. Otherwise, we won't be able to localize the receptor on the carrier. We have to pack the genetic material in certain particles which naturally will enter the place we need. But then we won't be able to ensure tropism. And this first approach, which is a non-virus mediated, is most often used in therapy by different small molecules. For instance, antisense oligonucleotides. First, it was used in urology for some diseases in neurology, inherited and genetically caused in case of myotrophic sclerosis. There are some antisense oligonucleotide drugs. The best known is the drug not for adult fall, but in case of pediatric spinal muscular atrophy. In case of adult forms that are quite new, there is a great genetic diversity, and by one drug we cannot have the correction of one gene and liquidating the consequences of all clinical form, because there is a dependence on mutation in the gene. In case of pediatric form of spinal muscular atrophy, there is one gene. So now we have 3 drugs to treat spinal muscular atrophy, because we know the genesis, molecular pathogenesis, the reactive developments of these approaches. Speaking about virus-mediated delivery, this is gene substitution therapy initially, so that is addition of additional copies of the gene that do not work and they operate in their natural cells. So many that was developed for autosomal recessive diseases where we don't have enough function of the gene, and more rarely that approach was used for autosome dominant conditions, whereas there is a synthesis of gene, but it has a certain defect. A virus-mediated delivery has certain advantages, so now it is one of the leading trends. First, this is a single delivery or delivery with a very large interval, once a year, once in 3 years, once in 5 years. That depends on the length of existence of virus in cells. If that is a virus-mediated delivery, we can ensure tropism, because certain viruses have tropicity to certain tissues, and then associated viruses were an absolute discovery for pharmacology and clinical medicine, and thus we can direct the virus to the place where we need, to liver tissues, to eye tissues, to the nervous system tissues, and that is rare introduction. Not once in a life, as a rule, the cells to a certain degree are divided or lose the genetic material, and we need reintroduction. In case of virus-mediated delivery, there are two possible approaches. We can inject virus to the circulation when it is injected to the blood flow and goes to the organs and tissues due to tropism to which it is targeted. These are most of the situations with which we work in gene therapy. The second approach, editing and introduction of virus in cells in vitro. That is a situation when we take cells from the patients, most of progenitor cells, and most of, it works in the target cells of the brain, progenitor blood cells, introduced in therapy of immunodeficiency. When we take the cells, we genetically modify them by introducing virus with a deficit of receptors, interleukin and immunoglobulins, and by means of transplantation of the bone marrow, they are returned to human organism and operate efficiently, having that copy of the gene. That is the transplantation of the bone marrow genetically modified by own cells. In the first case, it is rather safe. In the second case, that is quite efficient, but requires observance of rules, for instance, GMP rules, because the work with organism cleanse with subsequent introduction, that manipulation with the virus is strictly regulated, and now in our country it is not supported by legislation, because essentially these are biomedical cell products, and that law has not yet been implemented. For ensuring that viral delivery, one of the essential issues is what virus vector we're using. And the difficulty of using gene therapy was determined by failures to select the viruses that can be used. First, there are several classes of viruses. Some of them are integrating, that are introduced into the cell and are built into the human genome and are in the DNA structure. There is a difficulty because such viruses might not be safe, oncogenic, proto-oncogenic, and for some viruses, for lentiviruses, retroviruses, proto-oncogenic effect was demonstrated, so now they are not used so often. Another type of virus, non-integrating, these are viruses that get into the cell and remain in the cytoplasm. They are not introduced into the DNA, they do not introduce with human genes, and thus remaining in the cell, they ensure synthesis of the product without interfering with their own DNA. With these classes of viruses, first we use adenoviruses, that are aggressive viruses that can get into any differentiated cell. In cardiology, adenoviruses were the first attempts because we have a cardiac highly differentiated cell, and only adenoviruses can penetrate easily into the culture of the cell in cardiomyocyte. But that is not safe. These viruses have a certain degree of immune conflict, and that can cause immune conflict. So now we use another type, adeno-associated virus, that is quite similar. Mainly they are safe, and they are not immunogenetic, to a less degree of immunogenetic, and have a lot of serotypes that ensure tropism. And by these viruses, now we create certain drugs, for instance, for delivery of genetic material ex vivo. When we take cells from the patient, we modify them under immune deficiency and hemophilia thalassemia, because that is transplantation of hematopoietic cells, and we introduce them to the patient with the care of the gene that he needs. It is very convenient to have a situation when we have a possibility to introduce gene therapeutic drug locally. And one of the examples is atrophy of the vision organs, because eye and immune privilege organs, and we don't have any immune conflicts, and the drugs that are administered there do not get into the systemic blood flow. We can use a very low dose vector because locally it remains there. And in ophthalmology now we have several drugs for therapy of eye diseases that are genetically conditions for congenital dystrophy of the retina, because we administer these drugs once or with several repetitions, and they are not propagated act locally. The main advantages of these adeno-associated viruses is safety, then tropism that ensures by a greater number of versions and serotypes that have tropicity to certain cells, and the advantage is that it is easily to work at laboratory versus other viruses. We can transduce cells. These are versatile viruses. We can take certain genes from them, work at the laboratory, so we can make a lot of methylation that are stable and they have a stable virus condition. Now we use 10 serotypes in clinical plexes that each of them has tropicity to certain tissues, liver cells, heart cells, lung cells to a lesser degree. The type passes through the blood-brain barrier and can reach nervous cells, and we can develop certain drugs based on certain serotype of adeno-associated viruses. A breakthrough was the study in 2009 that showed that 9 serotype might penetrate through the BBB, and gene therapeutic drugs appeared for treatment of the nervous system. Now we have a whole line of such drugs that are actively introduced into the practice for treatment of spinal muscular atrophy. They are used on serotype 9, penetrating through the BBB. In addition to neurology and diseases of CNS, the drugs are actively developed that have tropicity to liver, that is a synthesis of different factors, hemophilia and mucopolysaccharidose, when there is a deficit of enzymes generated by the livers. Also the drugs for the muscles and for the eyes. A lot of serotypes of the atrophic muscular tissues. Most of them are tropic to the heart with certain advantage versus muscles or heart. So the diseases of neuromuscular systems are among the most attractive for development of gene therapeutic drugs. These structures are safe because from virus all the main genes are switched off that ensure its functions. Replicative genes, genes generating capsid, and the construction is built that we need for the built-in that we need for their therapy, so we can use them easily, and that is safe. Another approach for introducing cells might be the so-called minor RNA approach. It is not related to adenoviral therapy. It is generated independently due to other carriers that I am going to discuss now. And small minor RNA molecules, microRNA. This is the main pool of all RNA molecules in our organism. The more complicated is the organism, the larger the pool of that regulatory minor RNA that do not reach in their translation protein, but regulate in certain process. Using with those regulatory minor RNA, we might modify the work with our genes. There are several approaches used in clinical practice, and there are therapeutical agents based on them. The first antisense, antinucleated approach in the left part, that is introduction of single-chain molecules, which now are on matrix RNA by a certain chemical interactions. They destroy that matrix RNA. The second, introduction of microRNA. MicroRNA is short RNA synthesized in our organism. These RNA have a rather extensive tropicity. One microRNA has a lot of targets, so that approach is not extensively used for the gene therapy.
The siRNA is the low interference RNA, which is artificially synthesized in vitro. So actually, an siRNA is a microRNA synthesized in vivo. This means that we can introduce large RNAs, large molecules of RNAs, but considering that they are smaller and so bulky, they are very rarely used in practice. These molecules can be folded into a liposome, which looks like a small bubble. These liposomes may have different composition, but the principle is the same. They are bubble-like. And a second approach is the so-called GNAC molecule that can be captured by the cells and thus will be introduced into the cells. Those are the key two approaches on how to introduce the microRNA. In 2013, a Nobel Prize was granted for the microbubble transport and the cell communication via liposomes and microbubbles. Cells transmit genetic information via these bubbles by packing bulky information sequences into these bubbles. These bubbles are called today biomimetics. Those could be synthesized biologically or chemically. They can be equipped with targets or special receptors, but the principle is the same. There is a molecule packed into a tiny bubble, and this molecule carries the therapeutic substances. This actually promoted the development of gene therapy. So there was the first stage with lentiviruses, adenoviruses as carriers. And as of 2018, we can see that gene therapy has skyrocketed because the bubble technology was introduced. One of the first targets were different lipid failures and lipid mutations, including familiar hypercholesterolemias that impact and disrupt liver cells. And liver cells have many tropic carriers. Here you may appreciate the set of drugs based on either antisense technologies or low interfering RNAs that are packed and delivered just as I said. The pioneer experience, although, was a failure. siRNA was used for the B protein, which is one of the lipid metabolism regulator, suppressing the atherosclerotic risk level. So Camersen was one of the commercial drugs that could prevent myocardial infarctions and atherosclerosis. But in 2019, a large meta-analysis was performed with over 1,000 of patients, demonstrating that this drug is associated with liver stenosis. But today, there are different drugs. In 14 and 15, there were pioneering articles and papers demonstrating that the lower number of copies of the mutated gene can prevent coronary artery disease. Homozygous mutations in patients demonstrated lower triglyceride levels and thus lower cardiac risks. So there are two drugs based on siRNA or ASO. A different approach implies introduction of the missing receptor to the lipoprotein. This is the case when we introduce a new gene copy to the failing gene. So the drug based on PSCK9 is the drug of choice in this respect. We know that this molecule can synthesize antibodies and can suppress the trophy to the lipoprotein, the receptor trophy, I mean. Inclisiran is the drug of choice, which is safely used. It has a good safety profile and can be used in combination with the standard therapy to reach the target cholesterol level. So several clinical trials are going on, Orion 9, 10 and 11, but the results are pending. And lastly, molecular therapy with in vivo editing. The adenovirus is introduced to edit PCSK9 via CRISPR-Cas technology. In 2021, a paper demonstrated a positive effect of such therapy. And lastly, I would like to demonstrate another example demonstrating how microRNAs could be used to treat heart failures. So this can be administered to patients with myocardial dysfunctions, but also in patients with endothelial dysfunctions. The anti-MIR, known as one of the factors, can reduce the anti-pro-BNP level and fibrotic markers. So this approach is becoming more and more common, not only in lipid cardiology. So gene therapy is one of the most advanced technologies, which is based on transgenes to impact the pathologic target. Today, drugs are based on liposomes or adenoviruses. Due to the tropic features of viruses, the drugs can enable the therapeutic effect and target specific mutated cells in our body. Thank you for your attention.
Thank you. Thank you very much, Professor Kostereva,
for this overview of genetic gene therapy, new opportunities in cardiology, highlighting a wide spectrum of possibilities and also highlighting the most recent results in monkeys on PCSK9. Now we move to another presentation from Dr. Asya Talieva from St. Petersburg. She will touch upon familial hypercholesterolemia, specifically familial hypercholesterolemia in the Russian Federation, current state of the problem, challenges and perspectives. Asya.
Уважаемый профессор Катапана,
Distinguished Professor Katapana, distinguished Anna Alexandrovna, dear colleagues, today I'm going to speak about familial hypercholesterolemia, and I would like to refer to the review published in 2019, which refers to rare lipid pathologies and different phenotypes in various lipid pathologies. What is a rare disease, after all? This should account for 40 to 50 cases for 100,000 of the population. Today in the world, there are over 7,000 rare diseases known. As for hypercholesterolemia, we know that there are homozygous hypercholesterolemia and heterozygous hypercholesterolemia. The homozygous hypercholesterolemia and its incidence is 1 in 160 to 300,000 people. As for heterozygous hypercholesterolemia, it is more frequent, 1 in 200, 250 members of the population. And this is true for Russia as well and globally. As for the consensus document on rare diseases, we can see that the monogenic lipid metabolism disorders include familial hypercholesterolemia, and you can see the key genes that are associated with this pathology. In terms of percentage, the global papers show that 90% of cases is deficient LDLR gene, and you can see the distribution for the rest of the genes. However, it is very difficult to identify patients with other mutations. But apparently, and presumably, the ApoB defect is more frequent in Russia than the official statistics shows. So the LDLR defect is closely related to familial pathology. Today we are going to talk about familial hypercholesterolemia and actually why is it so important to diagnose this disease as early as possible. Let's look at the average time frame when patients develop CAD. So you can see this is roughly 55 years of age. The rare homozygous familial hypercholesterolemia, or in patients with untreated heterozygous familial hypercholesterolemia, manifest CAD by 35 years of age, so this is much earlier. As for homozygous hypercholesterolemia, those patients manifest CAD by 12.5 years of age. As for the contribution of low-density lipoproteins, we can see how the distribution curve changes, and in case of proved genetic mutation, we can see how different the low-density lipoprotein profile is. What is the impact of the genetic mutation on CAD depending on the low-density lipoproteins? Well, this is much higher. As for clinical scores, there are several scores used. The most common ones are the British scores that are commonly used in Great Britain, which is quite logical. Well, in Russia, for adults, we use the Dutch Lipid Clinic Network score, and the key idea is that we can diagnose patients and formulate the diagnosis without clinical tests if the patient has 6 and over points by DL&C score. The key issues in diagnostics and verification of familial hypercholesterolemia is the difference between clinical and genetic diagnosis. In red, you may see the patients demonstrating relevant data according to genetic diagnostics. However, patients who show over 6 points by clinical scores but do not demonstrate any genetic manifestations. And there is a different group of patients that clinically have the score lower than 6, but demonstrate genetic mutations. And as a result of sequential screening. So we do not know actually whether we are going to succeed with our conservative therapy in such patients. Let's look at some background and the evolution of treatment of patients with FH. There are two concepts, the earlier the better, and the other one, the lower the better. The latter one, the lower the better, is based on recent meta-analysis, including Fourier and others, who demonstrated good safety in patients with low-density lipoproteins. Let's look at some background at how or what is the history behind the target low-density lipoproteins values. Earlier, we used to have the target criteria of 100 milligram per deciliter, or which is equal to 2.5 millimole per liter. So today, such a study as the cholesterol and recurrent events study, CARE study, is something unthought of. Another trial, PROVE-IT-TIMI-22, demonstrated the positive effect of reducing low-density lipoproteins to 1.8 millimole per liter, which is incorporated into the guidelines dated 2016. And ultimately, the innovative era 2019 Odyssey outcomes and 4-year trial with monoclonal antibodies emerging allowed us to significantly reduce the low-density lipoproteins level to 30 to 50 milligram per deciliter. The key points in European and Russian recommendations dated 2019 and 2020 respectively is the risk stratification in FH. FH patients have very high direct cardiovascular risk. However, until 2019, we used classic risk evaluating scores, not the score scale. Today, we can classify these patients into high risk and very high risk groups and modify our strategy. The other concept, the earlier the better, is based on the Mendel randomization, which shows that it is not only the level of reduction, but also the time frame and the length of higher low-density lipoprotein in serum.
And arterial pressure. And thus it was shown that the earlier we started the correction, the less cumulative cardiovascular risk would be typical of our patient if at a certain transverse time limit, the LDLP in both patients are similar. That important is what preceded the increase of those values. As to the official recommendations, the statement of score that disappearance of total cholesterol 8 that is treated as a high risk, and the changing target levels of LDLP as to stratification of patients with FH. For any patient, the purpose is 1.8 or 1.4, depending on their high or low, very high cardiovascular risk. Another position concerns the following: similar levels on attaining the target values relative to patients with the FH, which are in the zone of primary prevention. Thus, we depart from the assessment of the primary or secondary prevention, and we proceed from the risk value, depending on which we choose the aggressiveness of hyperlipidemic therapy. In the structure of hyperlipidemic therapy, we'll not discuss it at length. In 2018, in Russia, officially, the Minister of Health approved clinical recommendations of FH, which essentially are a certain instrument for decision-making in clinical practice. And nevertheless, there are certain unsolved problems, mainly the concern the use of hyperlipidemic therapy for children. It is highly important because in Russia, pediatric network of lipid centers developed. Their purpose is to correct lipid metabolism in children. Thus, in official clinical recommendations, as well as globally, we say that it's necessary to treat heterozygous of FH from the age of 8 years and older. In the instruction to the drugs, we can see that that threshold starts at 10 years of age. Passing on to the current status on familial hypercholesteremia in the Russian Federation. Russia is a participant of the major European FH studies collaboration project. It wants to assess the frequency of genotypic and phenotypic features of the patients with FH. The register comprises more than 61,000 patients from more than 70 countries globally, and Russia takes an active part in keeping that international register. Several years ago, the National Society on Atherosclerosis set up the patients, the register of patients with FH that is called Renaissance. It was necessary to answer the question about the prevalence of such patients and how well we can correct the risks and attain the target values. Unfortunately, to consider the percentage of patients in secondary prevention zone is high. It is 45%. That means late revealing a diagnosis of the condition. Fresh publication of 2020 on the dynamic of indicators of lipid profile of the patient shows that, unfortunately, these data are relevant with the data of the international register, and the percentage of attaining the target values is low due to rigidity of low attainment of the target values. It is rather difficult to attain them without drugs of gene engineering, and the preferential drug provision is not optimum. The zone of primary prevention. Thus, the difficulties that we have from the scientific viewpoint, difference between clinical and genetic diagnosis, the need to use additional markers for precise stratification of the risk of the patient. The problem is adherence to the therapy, and to a lesser degree, to a greater degree, of the patients in the primary prevention zone. So one of the projects at the National Center of Personalized Medicine is to find genetic diseases. There is a theme on the study of phenotypical features of patients among the individuals with FH, and there is an in-depth register, which is not observation register, that is an in-depth register at which we assess genetic testing and detailed studies of the vessels, duplex scanning of carotid arteries, Achilles tendon, and we can relieve more sensitive markers for risk stratification in the zone where we have doubts concerning the tactics of treatment of the patient. Take into account the increasing importance of the index of coronary calcium in European recommendations and appeared in 2019. In the American recommendation, it was earlier. Thus, in collaboration with our Italian colleagues from Milano University, jointly with our Board of Radiodiagnostic, there is a sub-project under the register on assessment of coronary artery calcium on the patient. Thus, we are planning to have dual energy computed tomography to assess the morphology of the plaque with the same purpose of predicting the patients with a high cardiovascular risk, and for solving the problems of adherence to improve the control of LDLP. The European Society has launched a project, Lipid Clinics Network, for creating the vertical structure for implementation of the European standards and technical and clinical recommendations that we have discussed. And the Russian Cardiological Society is an active partner to the project. And now, under the auspices of Almazov Center, 4 regions have set up lipid centers involved in that network. They take active part in recruiting the patients and supplementing the register, and that register was considered as an instrument for the possible for people with confirmed genetic testing for preferential drug supply in gene engineering if necessary. And probably the final slide, the gap between the clinical recommendations and real practice dictates the necessity of new approaches in hyperlipidemic therapy, and I think this is the freshest publications that appeared 2 weeks ago. Were moving along the same trend as the scientific work from real hypertension, where today there is a combined therapy of the first stage, and the patients of a very high risk with high values of lipoproteins of low density. Such position for them might be justified in terms of minimizing the side effects and the maximum attaining of the target values of LDLP within a short period of time. I would like to thank you for your attention. Thank you. Thank you very much.
Please go.
Please, Professor.
Okay, thank you very much, Aziat, for this presentation. And we now move from the familial hypercholesterolemia in Russia to the familial hypercholesterolemia in Italy, and this will be presented. As you have understood, there is a strong collaboration between the groups. Will be presented by one of my closest co-workers, that is Dr. Manuela Casula, and she will speak and present us data about the experience on familial hypercholesterolemia registry in Italy. Manuela.
Please, can you confirm you see my screen?
Yes.
Okay. Okay, thank you. Thank you so much for this kind invitation and for the opportunity to describe our experience with the Italian registry of familial hypercholesterolemia disclosures. And I think we can start remembering that familial hypercholesterolemia is a common genetic disorder originated by mutations on genes encoding for proteins involved in cholesterol metabolism, and these mutations lead to an impairment in the activity of LDL receptors, and this results in an increase of LDL cholesterol levels since birth, with an accelerated atherosclerotic process and development of cardiovascular events, for example, coronary heart disease early in life. And as remarked in 2015 by our European Society of Atherosclerosis with a global call to arms, the familial hypercholesterolemia need huge effort to address the global burden of this disease. First of all, for the clinical burden of familial hypercholesterolemia, as we already heard, patients with FH have a higher risk of cardiovascular events, for example, coronary heart disease, cerebrovascular or peripheral vascular disease. It is true also if we consider comparable levels of LDL cholesterol, and this is due to the presence of hypercholesterolemia since birth. The second point is the underdiagnosis of the FH, as we can appreciate by in the figure comparing the actual number of patients identified with FH with the theoretically predicted estimates based on the frequency of 1 out of 500 patients. And as a result, we have also a problem of undertreatment of FH. First of all, this is a consequence of the underdiagnosis, but it is also due to a sort of treatment inertia by physicians that make little use of the most effective treatment options currently available to reduce lipid levels. And this features prompt a comprehensive and integrated effort addressing different aspects of familial hypercholesterolemia. And among the potential strategies, pathology registries could have a crucial role. And indeed, local and national at local and national level, the potential of these instruments are well demonstrated by results published by registries established for the longest time. For example, the Cascade FH registry in the US, or the SAFER registry in Spain, or the Canadian registry, but also the possibility of pulling together country-specific data allowed to obtain large-scale real-world data necessary to fill the gap in knowledge about this disease and to improve collective awareness. And this is our experience in Italy with the Lipigen network. Lipigen first is a network of lipid clinics that with the objective to share protocols to improve the detection and the management of patients with genetic dyslipidemias, to promote the genetic analysis and to increase awareness among physicians and patients. And this is our network of lipid clinics with a long-lasting experience in managing patients with primary dyslipidemias, and up to now about 50 lipid clinics are involved in the network, in the Lipigen network, accounting for almost 9,000 of FH patients. And the first step in the data collection in Lipigen is the clinical diagnosis, and it is based on the physical examination of patients, searching for classical signs of FH, for example, symptoms of xanthelasmas, the evaluation of the clinical history of the patient, mainly regarding the history of cardiovascular events, the evaluation of the family history of the patient, mainly regarding the presence in the family of hypercholesterolemia or premature cardiovascular events, and also LDL cholesterol levels. And the clinical diagnosis is to identify patients eligible for the confirmation of the genetic test. searching for mutation on the candidate genes. And the collection of data about, real data about patients with FH has dramatically increased in recent years. And this allowed us to appreciate the potential of the pathology registry in generating evidence and improving knowledge about the FH. First of all, the potential of the FH registries is the utility in the local adaptation of algorithms that support the clinical diagnosis of FH. For example, in our experience, we evaluate the performance of the Dutch Lipid Clinic Network score in a subsample of the lipigen cohort on about 1,300 adult patients with a genetically confirmed FH. And we were able to observe that the Dutch score failed to identify patients with FH in about 32% of these patients. This could be due to the presence of missing data, and we were able to observe that these were mainly due to data regarding the family history of these patients. But this was also due to the temporal trends, the secular trends of the epidemiology of some signs correlating to the FH, for example, the presence of xanthomas, as you can appreciate in the table, comparing the prevalence of xanthomas estimated in this cohort, evaluated between 1972 and 1978, compared to the prevalence of xanthomas in the cohort of lipigen. And we were also able to identify, as for example, the one in the slide, some potential other variants that could be added to the classical algorithm with the possibility of improving their performance in the diagnosis of patients with FH. A second point is the possibility to evaluate the correlation between genotype and phenotype. In our experience, for example, we selected among the lipigen cohort patients which were who were homozygote FH, presenting the same mutation on both allele of a single gene, compared to the compound FH presenting two different mutations on the two allele of a single gene, and double FH with two different mutations on two different candidate genes. And in the tables, you can appreciate a sort of trend in LDL cholesterol levels, moving from double to compound FH to homozygous FH. And this was reflected also in the prevalence of cardiovascular disease, or personal history of cardiovascular disease, and also in the prevalence of atypical signs of FH, as for example, xanthomas. The third point could be the possibility to explore the genetic base of this disease. This is we in the lipigen we were able to describe more than 200 variants in about 1,000 patients, mainly pathogenic or likely pathogenic variants, and mainly on the LDL receptor genes, of which 27 of these variants were not previously described. And we also tried to evaluate and study a recently proposed base for this disease, that is a potential polygenic base. And we applied the LDL score of 12 Smiths proposed by Talmud in 2013, And we were able to confirm the correlation between the LDL-C score and the LDL cholesterol levels in our cohort in the Lipid study, and also to confirm the higher value of LDL-C score in patients without a causative mutation in one of the candidate genes compared with mutation positive patients. Another possible area that could be explored with data from registries is the study of treatment response. In Lipigen, we are now collecting data about the follow-up of our patients, so we have not yet results, but other registries address this topic, and here you can see the results from the Spanish Safeguard registry, showing a poor level of treatment of each patient and a poor level of achievement of the LDL cholesterol goals recommended by the European guidelines, both in primary and in secondary prevention. And this was also confirmed by this data from the US Cascade FH registry, in which more than half of the cohort of FH patients did not achieve the LDL cholesterol goals of 100 milligrams per deciliter. Then a registry also allowed to study specific subgroup of FH patients. We recently focused our attention in Lipigen study on the pediatric population, and we were able to collect the data about more than 1,500 FH patients younger than 18 years old. And we classified these patients according to age classes. We were able to evaluate the LDL cholesterol levels in these different age classes, observing that overall younger patients, as expected, showed lower LDL cholesterol levels compared to older patients with FH. But we could also appreciate a wide variability of the LDL cholesterol levels across the different age classes. And this is a key point in the design of specific algorithm able to support the diagnosis of the disease in this particular population. A last point could be the possibility to identify cardiovascular risk factors in this specific population of FH patients, because we know that the classical algorithm designed to in order to evaluate the cardiovascular risk, for example, the European score or the Framinghar, cannot be applied to the FH population. And data from registry could suggest, could help to identify some additional factors that can help to stratify each patient according to their cardiovascular risk. And this is, for example, the experience of the Canadian Montreal FH score or the Spanish state-part risk equation. So to conclude what I briefly described are some possibilities, some aspects that could be addressed using real-world data collecting by different national registries and from evidence obtainable from pooling data from different national realities. And in this way, I think that pathology registry has proven to be a valuable tool to direct basic research, to guide clinical decision, and to inform stakeholders in health policy, with the final aim to improve the clinical management of patients with familial hypercholesterolemia. So thank you so much.
Aziat, please, would you like to take over and proceed?
Да, спасибо большое, профессор. Мануэла, большая благодарность за блестящую презентацию.
Manuela, thank you very much for such a fascinating presentation and for presenting the Lipigen registry results, which is one of the benchmarks in Europe in terms of its comprehensive collection of information as well as its scope. And it's so valuable that you also have pediatric patient, which is an indispensable, a crucial part of registries. Manuela, thank you, fascinating. And it's my pleasure now to give the floor to Dr. Vasutina, prospects for experimental biomodeling in atherosclerosis. Dr. Vasutina, the floor is yours, please. Thank you. Okay, distinguished colleagues, distinguished moderators, today I'm going to present my report on prospects for experimental biomodeling in atherosclerosis. What is biomodeling? Biomodel is a genetically modified animal with an artificially modified genome. However, I would like to note that, of course, there are not only genetic models, but also surgical, clinical models that can be used for personification. I'm not going to focus on the pathology and the genesis on dyslipidemia. It can be primary dyslipidemia, and we could be confronted with secondary dyslipidemia, which is developed based on the lifestyles or is associated with drugs. So we can also split our animals into two types: primary dyslipidemia and secondary dyslipidemia. And our approaches are going to be different. Dyslipidemia is quite often associated with hypertension, abdominal visceral obesity, hyperglycemia, insulin resistance, so on and so forth. Hypercholesterolemia is a biomarker of atherosclerosis and cardiovascular complications. Quite often, hypercholesterolemia is associated with elevated LDL-C, and there is a non-HDL-C cholesterolemia. We also have elevated triglyceride levels. To reduce the risk, we have to reduce the low-density cholesterol levels. So the pathogenesis of atherosclerosis is the emergence of an atherosclerotic plaque, which increases the permeability of vessels and propels the integration of monocytes. So these two stages are going to be our focus. We are going to look at fish models. On top of that, the vascular cells migrate into the intima and synthesize the cellular matrix. The cells become apoptotic, and thus there is higher risk of atherosclerotic plaque rupture. So thus this process has huge impact on destabilized atherosclerotic plaques. So mice models, however, are more prone to stable atherosclerotic plaques. Therefore, this model is actually unfit for humans, because in humans we see unstable atherosclerotic plaques more frequently. Overall, the biomodeling in animals is accelerated atherosclerotic plaque formation associated with the Western type diets. We should also note that the speed of atherosclerotic plaque formation is the key parameter in choosing the most appropriate model. So we can see several atherosclerotic plaques models. So we looked at different factors, including the cost of animals and the feasibility of using these particular animals to model the human process. On top of that, we also looked at the sensitivity of animals to the environmental factors and the type of diet. Mice, one of the most favored model to study atherosclerotic plaques, do not express CATP, whereas fish and rabbits do express CETP. Atherosclerotic resistance, which is demonstrated by wild-type mice that are highly sensitive to atherosclerotic, those are 57 black mice. So overall, mice show low sensitivity to environmental factors that promote atherosclerotic plaque formation. I would like to present the meta-analysis on different animals. Horizontally, you may see the time frame, and vertically, you can see the number of PubMed publications. But it is presented in logarithmic scale. As of lately, we can see elevated interest to fish models. However, PubMed demonstrates less than 100 papers on that topic, which means that this is a highly specific topic. The most prolific author is Danny Oreiro, who looks at zebra fish. As of lately, Danny Oreiro, a tropic fish, is very popular to study lipid metabolism. This fish has transparent embryos, and thus we can track different stages of embryo formation and track the formation and the evolution of bodies. Danny Oreiro, as a biological model, is frequently used in Europe, US, and some Asian and countries and Japan. In the first part of the 20th century, some background information. Diet-induced atherosclerotic models in rabbits were used. So rabbits were the most standard body. It is in rabbits that it was shown that high cholesterol level and high protein diet results in hypercholesterolemia. Different diets were demonstrated in rabbits that can induce hypercholesterolemia. Over that time, different types of diets were validated, and we use this data, we refute on this data. Now, in 70s, 80s, atherosclerosis was studied in lab mice. In 80s, cholesterol in serum and transgene technologies resulted in emergence of knockout mice, like knockout mice for LDLR or knockout mice for ApoE. On top of that, black mice C57BL6 were synthesized with metabolic disorders induced by diet. These mice are usually used to modify the background for genetically modified mice. Danio fish genetic modifications date back to early 80s. In 2000s, this type of papers has increased. The hypolipidemia model ApoC2 was developed. So scientists became more focused on Danio fish due to simplicity of genetic modifications and manipulations. But this model allows to study different disease processes like fatty liver, visceral obesity, disrupted intestinal lipid metabolism, cardiomyopathy, retinopathy, brain disorders. And on top of that is hyperglycemia-induced wound healing defects. Danio are close relatives to spine animals, and they are valuable as a biological model for extensive pharmacological and clinical screening. You can see that this model is quite comfortable to use, especially for screening. Danio fish can be easily susceptible to morphologic genome screening. One pair of fish can yield over 500 of eggs. Well, 200 of eggs on average, so thus we have 200 of eggs with identical homogeneous background, and this allows us to have a very powerful control over the process. These fish demonstrate non-specific embryogenesis. This allows us for phenotypical screening. What is exciting is that the fish is transparent, and we can use fluorescent colors and markers to do genomic screening. We can use high-resolution methods to investigate individual organs. However, there are other types of transparent fish like Casper fish. Overall, this is a very convenient model. Let's compare mice and Danio as biological models to modify atherosclerosis. So there are different factors. Some factors are more valuable for science, others are more valuable for practitioners. As a practitioner, I'm a vet doctor, would value practical factors more. So what is important is the simplicity of breeding, the specificity of genotyping, because in mice we normally use PCR genotyping.
In case of fish, they have a good survivability and good conditions. You can have genotyping by fluorescent microscopy, and we simplify the task. We might get a great more homogeneous sample, and the model is quite convenient. As to the simplicity of keeping, as to the rodents, there are several systems. The most popular system that is used everywhere, that is keeping in individual ventilated cells, and that is used for the animals of best conditional status. Thus, we maintain microbiological status of the animals. We simplify the work of the staff, but it is rather expensive. As for the fish, they are also contained in automated systems in aquariums or in tanks, and the main feature is water preparation. Naturally, they live in a liquid environment. Liquid environment has a trend to growth of different pathogens, and naturally it is necessary to maintain certain salinity of waters and conditions, but the systems both for mice and for fish are automated, so the conveniences are comparable. As to the simplicity of modeling, you can see mice that is below Dana, because for mice modeling is not characteristic using exposomal factor or medium factor. In order to model bright and express pathological process of the mice, it is necessary to combine the medium factor with genetic factor. We are not speaking about chemical induction. In case of Daniel-Rero, the situation is different. They are sensitive to the medium factors at different stages of their evolution. We can affect on larva and eleutherium embryo. That is an interim stage of development of Daniel-Rero when it is not caviar, but there is a sac, yolk sac from which it is nutrited. That stage is similar to invertebrates. That simplifies work with these systems as to the bioethical committees, and that stage also causes particular interest in the sphere of study of lipid metabolism, because in the yolk sac there is certain specificity of lipid metabolism. We can affect both on larva and eleutherium embryon and adult fish. We can work in different ways, starting from lighting. There are models where we induce the light action on caviar, and in chemical action it is rather easy to provide the action because most of reagents or active factors are ready to water, and there are some advantages. We can easily assess the number of consumed food. In case of fish, that is too difficult because they are not kept individually, and we cannot trace the food in fish with such easiness as for the mice. The next factor is ensuring control of microbiological status. That is a veterinary history, but that might be important for the research. We're working mainly with animals, rodents of SPF category that have microbiological status. We know what they have, what they don't have as to the pathogens. With fish, the situation is difficult because we don't have sufficient quality. We don't have sufficient labs that will study the microbiological status of fish, and liquid environment promotes the fact that we can easily bring something to the environment than for to the medium for rodents. So there are publications that say that it is difficult to control microbiological status of fish. As to similarity of humans in pathology of atherosclerosis, first, we have said that mice have a trend to forming stable atherosclerotic plaque. For some investigations, there is a good, but there is a trend that we are not interested in stable atherosclerotic plaque. That is a disadvantage. And atherosclerotic alterations in coronary arteries in mice are quite rare. Commonly, they occur in other vessels. As for disadvantages concerning fish, the main problem, that is one of the main problems that makes the researchers not use the model. We can say that that is polytropicity. We cannot use the aspect of thermoregulation in the lipid sphere of the man, but we have other factors that can be extrapolated naturally. Fish and mammals have different respiratory and reproductive systems, but zebrafish are quite good for studying early stages of the formation of atherosclerotic plaque, because the first two items, pathogenesis, are well pronounced in this model. As for the similarity with humans in lipid profile and metabolism, the main disadvantage of mice is that difference in lipid profile determines a certain resistance to the medium effect that might induce dyslipidemia. As to the fish that has a base default hypercholesteremia versus mammals, that is also a specificity of metabolism. Lipids prevail over carbohydrates as an energy resource. Fish express CTP, unlike mice, and they express leptins and receptors in the fatty tissues. As to the subjection to the medium effect, we have mentioned mice. As to the fish, a broad range of effect by the medium fish might be present. We can change different combinations, diets, and light. different chemical agents and a lot of mutations, but that is a different story. As to the mice, non-background mutation, light would not give such a pronounced picture. As to the genetic manipulation, both organisms are well studied. Genomes have been sequenced. Fish contain 70% of homological genes, autologous mice about 80%. Therefore, both models are good, but for mice there is a great number of protocols. For fish, several protocols are still being developed, but minus the specificity of embryogenesis or multiplication that makes these genetic manipulations. As to the research methodologies, it is also interesting. Daniel Herrera, at different stages of development, we are speaking about embryonal and transparent. We have great possibilities in imaging, in assessing the condition of the organs and development, evolution of the organism, the organs, and that's great. We also use whole body technologies and omics study based on specific architecture of the organism that can be done simply in case of fish. It is difficult to take blood samples. We analyze the lipid condition of the blood in mice. It is easier to take blood sample. As to the mice, it is good that they have physiological shows of disturbances that are well seen from the veterinary viewpoint. When we assess the animal, we can assess its state. It is not so easy with the fish. Greater access for the blood samples, for biochemistry, imaging systems are limited. But nevertheless, there is a great number of possibilities starting from CT and up to MR. So in principle, the indicator is good. And to conclude my presentation, I would like to focus on the following. These model animals are an instrument for translation of data which we obtain to the clinical practice. So feedback is important because between science and clinics. We have to determine the target substances that might be interesting to the clinician to model it using, for instance, Daniel Herrera, because modeling of certain phenomena is rather quick, and now science is developing, and genetic editing is made for Daniel Herrera quite actively and ubiquitously. So we ask all the clinicians to interact in that field. Thank you very much. Thank you very much, Marina Lvovna. Thank you for your wonderful presentation, because I think that most of the attendees were quite surprised to reveal that Daniel Herrera model, physiological and pathophysiological, doesn't have any advantages versus rodents. That is quite striking, because it's rather difficult to work with rodents. And it always seemed to us that atherosclerotic models with rodents are not possible for us due to some organizational problems. And in model Daniel Herrera, they are quite surprising. They're quick, and we can produce genetic modifications. I think that for everyone, it is quite clear that they are far away from lipid metabolism, atherosclerosis. That opens good possibilities for our young collaborators, particularly. They can be used as possibilities to create genetic models and atherosclerosis. Their models, I think, will complete our section. We are within the time limits. I think that we had a very interesting section that was quite versatile, with population screens, with the possibility of studying familial rare forms with genetic approach and basic molecular therapeutic at the level of the animal models. And I believe that this section in atherosclerosis from clinic to genetics should be done traditional, and probably we think with Alia that we might have some youth school in autumn to educate the young experts in these issues. Thank you very much. Professor Katabana, do you have any wish to add a few words, a few comments to conclude?
Just to close, to thank all the speakers for the excellent presentations. And as we are quite out of time, I wouldn't take more time. Thank you to everyone, and see you during the next part of the meeting.
Спасибо большое, коллеги. До свидания всем.
Thank you. Goodbye.